I think that we can get started. So please, I would like to welcome Mary Estes. She's a professor at Baylor College of Medicine, and she also holds the current endowed chair of molecular and human virology. She is doing an excellent work on researching and trying to understand the viral infections on the gastrointestinal tract, especially rotaviruses and noroviruses. And I think that everyone here is really interested and excited about her talk. So please, whenever you are ready, Mary.
Okay. Share screen. And that's at the end. Okay, how do I go? Well, top. You seeing that? Is it full screen?
No.
Okay, now. So now is it full screen, or I need to change the?
I think that you need to change it.
Like that?
Yes, that's perfect.
Okay, great. Well, I'm delighted to have this invitation to speak at the Gladstone and to share some of our work on our GI viruses, and the cultures that we use are these human intestinal organoids, or we call them enteroids, actually. And this first slide, let me see, shows you some pictures of the virus. This is a negative stain EM of norovirus. That's really what I'll be talking about most today. This is a crystal structure of a virus-like particle. We don't have enough replicating native virus with genome in it to have that structure yet. But the particle is made predominantly of one capsid protein, shown here. It's color-coded like this. It has a protruding domain and then a shell domain that makes up the shell. And on the right, these are two different, this is a picture of an intestinal organoid. And you can see that these are quite remarkable. They have a glycocalyx, and under some circumstances, they do make villi. So I like to talk about gastroenteritis because not everybody, and particularly people in the United States, probably don't think about it much. But one picture is often worth many words. And this is still a very important problem globally. It's been estimated that every day, 200 million people on Earth have gastroenteritis, and the amount of water equals the amount of water that's flowing over Victoria Falls in Africa in 1 minute. So this, you can imagine, is a big problem. And this slide gives you just some overview of the numbers. There are about 3 to 6 million children that die every year from infectious gastroenteritis, and death is from severe dehydration. This is a child that has severe dehydration. There are many causes: viruses, bacteria, and parasites, and I won't read these all. The two that my lab studies are rotavirus, which is a double-stranded RNA virus that I'm not really going to talk about today, and then the other one is norovirus, which I will talk about. But you can see there are many viruses that affect the gastrointestinal tract, including SARS-CoV-2. This is a fairly simple slide, but I just want to be sure we're all on the same page. And when we talk about the intestine, it shows you different segments of the intestine, and the viruses that I work with are actually only infecting the small intestine. And it's got different, very specific different segments. In the small intestine, there's the duodenum, jejunum, and ileum. And those all have specific functions that help us. The duodenum helps us chemically digest food. The jejunum is important for the absorption of nutrients. The ileum absorbs leftover nutrients and vitamin B12 and bile acids, which are recirculated to the liver and then come back down through the bile duct to the duodenum. And the colon absorbs water and, of course, has lots of bacteria that break down waste and so forth. And I'm showing you this because we will be dealing with, we have made cultures from all these different segments of the small intestine and the colon to study our virus. This slide shows you a little bit of an overview of the intestinal epithelium. The intestine is this really very complex tube that has both muscle in the bottom, and then it has crypts at the bottom and villi that come up off the muscle. This is a more detailed structure. This would be a crypt, and in the crypt, the stem cells are found at the base of the crypt. There's still some question about exactly what is there more than one stem cell, but these then move and become transit amplifying cells, and then differentiate and move up onto the villus, where they become all of the differentiated cells that are finally in the villus. And those cells include enterocytes, goblet cells, enteroendocrine cells, Panus cells, and actually tough cells, which are not on this slide. When this epithelium is one of the most active epithelia in the body. It's renewed about every 4 to 5 days, so it's constantly undergoing renewal. And the system that we're using to study our viruses is called, it's an ex vivo culture system. We're making tissue-derived organoids, or also called human intestinal organoids, and that's what we've called them, so I'll call them that in this talk. And we make these cultures by getting a biopsy or surgical specimens from someone that may be undergoing a procedure or having surgery. We then take a piece of tissue, and in this tissue, this shows you an H&E of the structure of the intestine. We isolate crypts. We put those in culture with growth factors, and within 4 or 5 days, you begin to see little enteroids being formed. And these little 3-dimensional cultures have a lumen in the middle, which would be like the lumen of your intestine. The stem cells are on the outside. And that's how we keep our cultures growing. We're growing them in 3D with growth factors. Ultimately, we then take these, we break them open, and then we plate them on 96-well monolayers for most of our infections. You also can work with them on transwells, so you can have an apical and basolateral surface, and you can invert them as another way to do infections. But we haven't done that, but Manuel at Stanford has set that system up beautifully. This technique was pioneered by Sato and Clevers in the Netherlands, and these cultures are non-transformed. And they can be indefinitely passaged. We're still working with cultures that we made in 2012. They make all the epithelial cell types. They're physiologically active. They exhibit circadian rhythm, and they retain patient specificity. And I'll show you an example of why that was important for our virus. And they retain the complexity and organization of the epithelium. So we've been using these. Once we set the system up, we actually set it up because we wanted to try to grow norovirus, and that's the story I'm going to tell you today. But we're now using them with collaborators to study a variety of microorganisms. So we've studied rotavirus and norovirus. Other people here at Baylor are studying enteroaggregated E. coli, commensal bacteria, and parasites in the cultures. And our overall goal has been to define mechanisms of host restriction and pathogenesis of microbes that cause human disease, and to investigate human host genetic susceptibility to enteropathogens. So I want to tell you a little bit of overview of human norovirus, which is shown here. This was a virus that was first actually identified from a 1968 outbreak of winter vomiting disease that occurred at an elementary school in Norwalk, Ohio. And it was first visualized by immune electron microscopy in 1972 by Al Kapikian at the NIH. So he took serum from someone who'd recovered from the infection and mixed it with stool and put these particles under his electron microscope, and lo and behold, he found particles. This was really a remarkable breakthrough. At that time, everybody really thought that gastroenteritis, which is vomiting and diarrheal disease, was caused by bacteria or parasites. And so this was the first virus that was visualized. And then rotavirus was discovered later. And this shows you the picture of the elementary school in Norwalk, Ohio, where that outbreak occurred. For many years, we didn't know what kind of virus it was. Some people thought these particles might be phage, except they were infectious, and Al Kapikian did volunteer studies with stool filtrates and showed that they were infectious in people. And we got some virus from him, did some volunteer studies, and we cloned the genome in our lab in 1990. And this turned out to be a calicivirus. It's about a 7.7 kb single-stranded RNA genome. It makes a subgenomic RNA that makes the capsid proteins VP1 and VP2. And either VP1 alone or VP1 and VP2 will self-assemble into virus-like particles. And we use those for many of our studies. So this is the leading cause of foodborne illness and of epidemic acute gastroenteritis worldwide. It's estimated to cause about 200,000 deaths globally every year and to result in greater than $60 billion in costs for direct and indirect health costs. It can cause chronic disease in immunocompromised and transplant patients. And if you get chronic disease, there's no way to treat you. And people can be sick for years. Some people will end up dying from it from dehydration. At the moment, there's still no licensed vaccines or therapeutics for prevention or treatment. There is several vaccines are in clinical trials, and one that we started is now being developed by Hilovac. It's in a big field trial at the moment, and the end of this year will be a big year to know whether it really worked or not. All the data so far says it's a very safe vaccine and it's immunogenic. It induces antibodies. And the virus was non-cultivatable for many years until we cultivated it in 2016. To give you a little more background about the viruses, they're genetically diverse. This shows you the viruses in red here are the genogroup 2 viruses. The prototype virus was caused of genogroup 1. And then there are genotypes, which are shown here. So it's G11 was the prototype virus. Most of these viruses infect only humans, and there are a few of the virus here that will infect animals. But at this point, there's really not been any evidence of cross-species transmission. The G24 virus, which is shown here, is a pandemic strain that has many variants. And this graph here shows you that there have been waves of different variants of this virus since 1995-96, and there's one additional variant that appeared this year in San Francisco that's not, I need to update this slide. And so this virus is emerging as many viruses do. The G24 strain is the pandemic strain and causes about 40 to 70% of the diseases, and there are these new variants. G23 is another virus that I'm going to be talking about today. It causes about 5% of outbreaks and 25% of sporadic cases, particularly in young children. And so once we got the virus growing, one of our questions was, are there strain-specific differences of these 2 viruses? And so we'd been trying for many years to try to grow this virus, and I'm a proponent of believing that new technology really helps solve scientific mysteries. And so when we read about cleavers being able to make organoids or enteroids, we decided that that was something we should try for this human trophic virus. And so we were successful, and our first paper was published in Science in 2016, which seems like it's a long, long time ago now. But I'm going to tell you what we've learned since then. Our approach to growing the virus was we took 10% suspensions of stool. And this shows you some pictures of particles that can be present in stool. We filtered them. We put them then on our cultures. And this shows you some of the heterogeneity of the cultures. So here, this blue is showing you MUC2, this is showing a goblet cell in the cultures. They're polarized here. These are cultures that are on transwells. And what we did was we infected the cultures apically. We started with monolayers. We looked at genome replication. We did immunofluorescence. We did flow cytometry, electron microscopy, looked for cytopathic effect, and stained for double-stranded RNA for replicating RNA. And this is typical of the kind of data that we get. And I'll tell you now, one of the things that's still frustrating is although we've got the virus to grow, we do not have it indefinitely passaging. So we're still working with stool. So one of the things we have to do is have a constant supply of stools coming in that contain virus. I can tell you that's not so easy. But what we did was we took 3 wells in a 96 well plate. We infected them, well, actually, we infected 6 wells. After 1 hour, we took 3 wells, washed off all the virus, and then extracted RNA. And we got some background amount of RNA that was present just after absorption. And then we took another 3 wells at different time points. This shows 72 hours. This is with the G24 pandemic strain. And then you can see we're getting increases in replication over time. And you can see that with, these are different variants of the G24. And from the beginning, we could already grow 4 variants of G24 virus. And I'll tell you later why it was important. And we were lucky that we started with this virus. We did immunofluorescence, and we could see for the capsid protein, and we got probably 40% of the cells were infected. And when we did immunofluorescence on the trans wells, you can see the capsid just underlying the apical surface of enterocytes. We could see particles being released if we concentrated them. And one thing that we weren't expecting, although actually there was some data suggesting this could have happened, is there were some tinier particles. So these would be T=3. If you're a virologist, you know what that means. And these would be some T=1 particles that don't have genome inside them. I say that we shouldn't have been surprised because in stools, there were 2 different sized particles. I always thought that these little particles were probably phage. But the fact that they're made in the epithelial cells say that they must be a byproduct, at least, of the infection. And from the time where we started that we could only grow really G24 viruses and G23, or maybe I think initially in the science paper, we maybe had 4 viruses. We can now grow 15 different viruses. And we know that the virus replicates in cultures from all segments, in these organoid cultures from all segments of the small intestine and the duodenum, jejunum, and ileum, but it does not replicate in the colon. And that's, we know not only from cultures made from different people, but we now are making a bank of cultures from an individual organ donor where we'll get all these cultures from a single person. And that's important because it's not a genetic problem that it's not growing in the colon. I've mentioned we cloned the genome from doing volunteer studies. And from those early studies, we ended up finding, this is a picture of some of our volunteers, and Anton Opekin was a physician assistant that helped us give cocktails to people on a Thursday evening. We had infected 51 people, and we found there were some people that got diarrhea or vomiting in both. And we discovered that these people are all people that are known to be secretor positive, which means that they have a functional fucosyl 2 transferase enzyme that puts fucose onto mucosal surfaces, onto proteins or glycolipids. And that turns out to be an initial binding factor for noroviruses. There were other people that did not get infected, and those are known as non-secretors. These molecules are also called histoblood group antigens or Lewis antigens. So what we found was that there are some noroviruses bind to multiple histoblood group and Lewis antigens, allowing infection of more people. The pandemic strain only infects secretor positive individuals, and the non-pandemic G23 strains infect some secretor positive, but they also infect some secretor negative individuals. So we went on to ask if our organoid cultures reflect this genetic susceptibility. And so we made 3 different cultures from secretor positive people. And we had 3 cultures from secretor negative people. And then we infected them with the pandemic strain. But what we found was that all of the cultures from the secretor positive people grew the virus. This is showing now fold increases from 1 hour, and all the cultures from the secretor negative people were resistant, just like in people. So these are really reflecting people's genetics. An interesting result we then went on and made knockouts. So we took our J2 line that was secretor positive, and we knocked out the FUT2 gene. We wanted to know, was this both essential and also just sufficient for virus replication? When we knocked it out, we saw this is staining UEA1 that stains the fucosylated molecules on the cell surface. When we knocked it out, we saw that that was gone, and in fact, we could see some fucosylated molecules inside the cell. to my knowledge, nobody else had actually ever seen this before. We have not gone on to actually characterize what these are, but that's something that's on our list too. We then took the J4 strain that was resistant to virus, and we knocked in FUT2, and you can see we now get that histoblood group antigen, the fucosylated molecules on the cell surface. And then we did infections. So in the J2 wild type, we got replication, and in the knockout, we didn't get replication. And in the J4 non-secretor, we didn't get replication, but when we knocked in the virus, we did get replication. So for this virus, FUT2 is really both necessary and sufficient for infection. We then looked at a couple other viruses, which she had growing. G17 is another virus that has crossed the world, and it behaved like the G24, and the prototype G11 did the same, that in the knockout, it didn't grow, and the knock-in, it did. But the interesting thing that we weren't not really expecting, but in fact, we found the G23 virus does continue to grow in the knockout line. Actually, we should have expected that because there are some G23, the viruses do grow in some secretor negative individuals. So this again reflected that. So the G23 virus, it's not essential for the replication of this virus. We did some more experiments. I've just told you we had G23 growing, but we didn't have it growing initially. And what we found was that we actually need to mimic the intestinal environment of these cultures to get some strains to grow. So for the G24 virus, we had grown it initially without bile. The G23 virus, if we'd started with that, we would never have moved on to think that these viruses could grow in these cultures. But we've been trying to figure out why some of these viruses didn't grow. And I went over and visited my gastroenterology friend, David Graham, he'd done a procedure that day and handed me a vial of bile. And we put that on this culture, and lo and behold, the virus now would grow. And when we added bile to the G24, it just enhanced replication. So this was another strain-specific requirement for replication. The G23 requires bile, or it turns out that it requires hydrophobic bile acids, and I'll tell you how in a minute. And we found that this is affecting virus entry, and we went on to try to explain that. So what is bile? I can tell you I didn't know a lot about it when I got that vial. It is made in the liver and stored in the gallbladder, and then every meal that you had, it's deposited into the upper small intestine, into the duodenum, then goes through the duodenum, and then it will be reabsorbed in the ileum and go back to the liver. So there's this interhepatic circulation of bile. And I've told you before, it absorbs nutrients from the stomach, and it contains bile after a meal. The major components are bile acids and other components, and we found two components that were important in bile for helping virus grow. One was bile acids, and the other was ceramide, which is a lipid. So to discover that, what we did was we took our human bile that helped the virus to grow. We used a drug called cholestyramine that's used to treat humans. It's one of the older drugs, and it will deplete bile of bile acids. And so we took the bile, treated it with that. You could see we depleted the bile acids. And then when we used that bile acid depleted bile for replication, you could see we got a dose-dependent decrease in replication compared to adding untreated bile. And so this actually raises the question of whether there are these bile acid sequestrants that are used clinically for a variety of treatments, and maybe they could be used to treat chronic human norovirus disease. I haven't gotten anyone to try this yet, but they are approved drugs, and I think I would try it if I was a clinician and had a chronically infected patient. We went on to try to find what were the bile acids in bile that might be important, and we found that the hydrophobic bile acids are ones that enhanced infectivity. There was one UDCA that did not enhance infectivity at all. And we went on to use now GCDCA and our other replication studies with G23 and other viruses because it's very soluble and it's not toxic to the cells, so it's easy to work with. We went on to try to understand, well, what was the bile acid doing to allow G2/3 viruses to infect? And to look at that, we used a fluorescent dye called FM43FX. And this is a dye, if you just put it on cells, it stays outside. But if you have endocytosis that occurs, if you have internalized compartments, it will now fluoresce. And we were surprised, but we discovered that bile acid induces endocytosis. To my knowledge, that had not been shown before. I don't think people ever thought about it. But here, this shows you that when we added just the bile acid, no virus at this point, you see that dye is inside the cells. And then we went on and we added fluorescently labeled virus-like particles. If we just put those on untreated cells, no bile acid, nothing happened, they didn't get in. If we put them on with the GC-DCA, which is now inducing endocytosis, we got virus inside cells, and you can see this is quantitated here. And this is now looking at the expression of the capsid protein, VP1, and you could see that it was expressed in the presence of the GC-DCA. So this is really letting the virus sneak into the cells. We also found that bile acid actually generates apical ceramide, and this is the molecule. There's frequently a lot of sphingomyelin inside cells that can be on the cell surface. When we added bile acid, this is a specific antibody to ceramide. We saw that we were now inducing ceramide on the cell surface. When we added ceramide by itself to cultures, we could get the virus to grow. And when we added ceramide and the bile acid, we got a synergistic effect. So the combination of those was important for replication. So ceramide also increased it. And this work really led to discovery of some new virus uptake mechanism that I am not aware of any other viruses using. And it also told us something about bile acid biology. And one of the new things we learned was we wondered, well, how is this bile acid, hydrophobic bile acid, getting into cells? It turns out that there is a G-coupled protein receptor called S1PR2 that binds to bile acid, and it then can induce a variety of effects in cells. This receptor was known to be present in the intestine, but very little is known about what it was doing. It's certainly in the liver and important there for signaling with bile acids. We could show that we had this receptor in our cultures, and when we went on and looked, there's a specific inhibitor called JTE for S1PR2, and when we added this inhibitor to our cultures, we get a dose-dependent decrease in replication. There have been some questions about whether there are off-targets effects of this inhibitor, and we found a second inhibitor, and we see the exact same thing. So this receptor is really important, and the bile acid binding to it is important for this infection. And we've also just recently found an agonist that can activate the receptor. And so, but just using the agonists in the absence of the bile acid also induces, lets the virus grow. So another hint that this is important. And then we tested whether the JTE would affect G2/4 replication, and we found it does not. So we don't know how the bile acid is enhancing G2/4 replication, but it's not just letting it get into cells. So I'm just going to show you our model now, what we think is going on with G2/3. So we think that the bile acids act on cells. They don't, we have a lot of, and they're acting early in infection. We did time course experiments. They don't act through detergent effects, and they're not acting through the classical FXR or TGR5 signaling. Instead, they act through this bile acid receptor, it's S1PR2, to induce rapid uptake and release of virus. And then we've gone on and shown that bile acids are required for a number of other strains, G11, G217, and a couple others that are not listed here. So we think what happens is that the bile acid binds to the receptor, it lets the virus get into cells. We think the virus is probably, and we think there is a proteinaceous receptor that is also present in lipid rafts, and that's a receptor we're trying to identify. Maybe that's S1PR2 is that receptor, but we've not been able to prove that yet. but the virus gets in, it goes into endosomes. We know acidification of endosomes is important, and we think that that can probably cause a conformational change in the capsid, which will release the genome into the cytoplasm. We also found that virus replication is blocked by acid-sphingomyelin, this ASM enzyme. And this is an enzyme that's important. It's in lysosomes, and it will bring lysosomes to the cell surface where ceramide can be made. And so having ASM come to the cell surface will generate ceramide, and that also enhances the replication of these viruses. So more recently, it's been discovered that, okay, let me see. I guess I have to turn that off. Let's see, I don't know how to actually turn this off. How do I do that? Okay, maybe I can, let me go back, sorry. Recently, a new structure of the virus done by Dr. Prashad at Baylor showed that this capsid is actually quite plastic. As you can see here on the right, the capsid moves. And the structure we'd always known about before was the structure, the shell domain and the protruding domain in a closed structure. And this new information shows us that actually there is a raised conformation that also occurs. We're wondering now if, in fact, this may be important for entry of some of the viruses. We know for sure that the pandemic virus, the G24 viruses, will undergo this kind of conformation. So we had some information about how the G23 bile acid dependent viruses got into cells, and we went on to look to see how the pandemic viruses get in. And this came up with data that was really a big surprise, another big surprise to us. This virus has lots of surprises. So for a lot of these experiments, we worked with our virus-like particles. And we found if we just added the virus-like particles onto the cells with this FM43X, the virus-like particles are actually inducing endocytosis all by themselves. And they were inducing membrane wounding, which we showed by showing also PI staining appeared with putting the G24 VLPs on. This didn't occur with the G23 VLPs. And if we looked at quantitated this, the pandemic VLPs would cause this endocytosis. We put just the P domain on and it didn't do it. So you really need the entire capsid shown here to get endocytosis of these viruses. And some of the other virus VLPs from the G24s will also get in the same way. But the bile acid dependent strains did not cause this. And so I'm not going to go through all the data, but what we ended up showing was that G24s induce membrane wounding and repair mechanisms that lead to a click-mediated virus entry. And there have been components of this reported before for the way other viruses gets into cells, but to my knowledge, this is putting these together for this particular virus is something that's also quite unusual and new. So I'm now going to shift gears a little and talk about, well, how do the cells respond to infections with these viruses? So we did an RNA-seq experiment on infected jejunal enteroids, and we used for this both infectious virus in stool, and we use gamma radiated stool because we're infecting with stool still, in case there are any other components in the stool that might be causing effects on cells. When we looked at the main upregulated pathways, it was interferon, not unusual, not unexpected for a virus, and cytokine signaling, which we didn't really know what that meant at that point, but we still don't completely know. You'll see that. But what we found was that certainly there were interferon lambda was the main interferon that was induced, and then there were other downstream ISGs that were induced. We made a STAT1 and MABs knockout. We also made interferon alpha and lambda receptor knockouts. And much to our surprise, we were expecting now this was going to help this G2.4 virus grow. It could care less. So I think this is an indication that this virus has lots of ways to antagonize the innate response. But when we put the G2.3 virus on, at least in the STAT1 knockout, we got enhanced replication, which was exciting to us. I can tell you the graduate student that did this, he did the G2.4 first and was really depressed. He didn't know what he was going to do with his thesis. But when he got the G2.3 result, he was excited. And when we looked in by immunofluorescence, we found in the wild-type cells, I mean, neither of these were really spreading very much, but the G2.3 virus now was suddenly spreading for the first time. We'd never seen that before. So these are, again, strain-specific differences in restriction by the host response. The G2.3 is restricted, and if you lift that restriction by making some knockouts, you could get spreading. Our disappointment was that we still couldn't get indefinite passaging. So I have a new graduate student who is really trying to understand what factors continue to rescue virus and might lead to passaging. And the way we've approached this was use comparative transcriptomics again. She's now using gamma and G2.3 virus in the HIEs, and then on our genetically modified HIEs. And this was an interesting result. We expected we wouldn't have as many ISGs, and in fact, we have very few. So we think, and she's now working on trying to decipher whether OASL may be one of the remaining ISGs that's still restricting this virus and possibly spreading. Maybe her C2 is. I was a little surprised to see these chemokines coming up in these epithelial-only cultures. And then in the STAT1 knockout, I wasn't aware that, at least in humans, that these are made in a potentially in a STAT1 independent fashion. If any of you know about this, let me know. You can educate me because I'd like to learn more. And we've gone on to do more comparative transcriptomics. I'm not going to show you all the data. I'm just going to give you a summary slide of where we are. So what we found, that there are all of these differences. The G2.3 is requires bile acid. It requires bile. It requires acid sphingomyelinase. G2.4 uses a different way to get in. G2.3 is not totally dependent on the FUT2 gene. G2.4 is, and we get enhanced replication here, but no change here. So we're now looking more at transcriptomics, and you can see, this doesn't list all the genes, but you can see that these are inducing very different responses. We think that G2.4 may be, and we have some evidence that this may be affecting not transcription, but actually translation of some of the proteins, and that may be how it's antagonizing some of the innate responses. But I don't have complete story yet, so we're working on that. And our big question is, what are the putative co-receptors? We have the S1PR2 as a potential receptor for G2.3. We now have some candidates for G2.4. We've done a mass spec of all the fucosylated proteins on enteroids, and that's just recently been published. We're assuming, we could be wrong, but we think that this probably is a fucosylated glycoprotein. And so we're going through now a list of proteins that we've got to look for the co-receptors. So I'm winding down and want to end with what are some of the applications that we've used for the system now that we have it. We can measure neutralization with serum antibody, which is useful. And in fact, we've shown that the vaccination with the VLP vaccine can induce neutralizing antibodies. And some of those are neutralizing more than one genotype of virus. So that's, I'm hoping, good news for the possibility that this VLP vaccine might actually work. We've been able to use it to inactivate virus. So if we gamma irradiate the G2.4 or G2.3, it doesn't grow. If we heat and activate, we can inactivate the virus. If you go up to 60 degrees for various lengths of time. That's important if you're traveling and eating food that might have norovirus in it. If you eat hot food, you may protect yourself. We did have one original strain that caused cytopathic effect. Unfortunately, we haven't gotten another one, and that could be neutralized by adding this neutralizing serum. And we've been able to show that at least one product that's a hand sanitizer could completely reduce infectivity. And this was great news. This is one that's used actually in surgical suites for sterilizing things. It's actually not yet on the market for trying to treat with infectious diseases. And we've known before that ethanol does not completely inhibit this replication. And finally, we've now looked with Prashad, found some, and other people have found antibodies that neutralize the virus. And what we're discovering is that there are different mechanisms that the virus can be neutralized in. This is the place where the histo blood group, the carbohydrates bind. And in this case, the antibodies that neutralize are binding and blocking histo blood group blocking, but they're also blocking up here at the tip of the P domain. Here's a different antibody that's now binding down lower, and another one that's binding in a different site as well. And this is one with those are all G24. With G11, there's another one that's binding and blocking the histo blood group antigen binding site. And then this is a fourth way that you can get neutralization. So this one is an interesting antibody because it was made from a person that had been vaccinated with the VLP vaccine that's being developed by Hillevax. And we know that, and this is a semi-conserved site. For some of these others, to get them to bind here, you've got to have the raised conformation of the particle. So I think hopefully I've convinced you that these cultures, I love this paper by Hans Clevers and Sato that looked at their view of how these organoids could be used. When I read this paper, I got very excited. And for me, it just was calling out that these were cultures that should be used for microbe-host interactions and pathogenesis, cultivation of previously non-cultivatable change. I hoped at the beginning, but now that's true. And other non-cultivatable organisms have been grown in these cultures. And then for the development of antimicrobials. And I think that these are certainly transforming enteric virology. This is from a review that we wrote a couple years ago, and lists. We've looked. It's helping us understand pathogenesis. We can now do neutralizing antibodies to help vaccines. We can look at genetic susceptibility and really learn a lot about these viruses. That's true with both rotavirus and norovirus. And now these organoids are being used for lots of different viruses. The brain organoids are made from a different technology using iPS stem cells and then differentiating them into brain-like cultures. But all the rest of these can be done in these tissue-derived cultures. And so we've done work with a variety of different viruses, and others have too, liver organoids. And of course, they're being used at your institution by Dr. Ott's group. They've been used. We're making nasal organoids that are very easy to make. You don't have to get a biopsy. You just brush inside the nose and you get stem cells out and can make those. And they're used for RSV and SARS-CoV-2 and could be other respiratory viruses. I think the future of this is making co-cultures with immune cells. People always ask, well, you're missing the immune cells from the gut. And we now have a system that we can add macrophages. What we found was actually M1 macrophages tended to kill the cultures, but M2 made them have a higher height. We're beginning to try to co-culture with neurons. This is a co-culture system from mouse, where you can see we've got a neuron perhaps wrapping around an organoid. And we have now an anaerobic chamber where we have an oxygen gradient, and we can infect apically. This would be anaerobic up top, aerobic down below, and you can use that now for doing co-cultures with bacteria. And there are lots of things to be done with these organoids. We've now made ones from children, actually pediatric or infant organoids, and it turns out that they're more susceptible to infection with norovirus and other viruses than the adult organoids are. They have a lower innate response is part of the reason for that. If you make organoids from people with different metabolism states, they behave differently, even for nutrient uptake. We've set up an inter-organ communication system between the intestine and liver that can be used for studying that system. And they're beginning to replace animals for many applications. The FDA has now said that you don't have to, for some pathogens like human norovirus, that you could potentially use organoids instead of animals for getting approval for some drugs. And you still would have to do toxicity experiments in animals. They're important. You can use them for zoonotic infections. You can really make them from any organism that has an epithelium. We're studying these multi-interactions, and it's really been fun because we're clearly an interdisciplinary team now, working with new bioinformatics, the kind of teams that you have at the Gladstone. And there's some cool organoids out there. There were active replication of SARS in bat organoids, and then Cleaver's group is always doing something creative, and he's made snake venom organoids that make, or organoids from snakes, yeah, glands, and they make snake venom, which I just think is amazing. And the group at Stanford has made organoids from tonsils, and they've modeled the adaptive immune response in those organoids. So I think there's lots to be done yet with these kinds of cultures. So I will end here. My group is really, it's the people that do the work that makes the difference. Okay, I have to learn. There we go. And our viruses, in the last couple of years, we've learned they're a lot more flexible than we knew. The people that have done most of the work, Suki Murakami did the bile acid work with Victoria Teng, and they've now left the lab, gone off to other future careers. Roy Lin did the initial single cell sequencing experiment. Gurpreet and Janam are now working at trying to understand the antagonism mechanisms in the organoids. And we do have an organoid core at Baylor College of Medicine that we make lots of organoids. We have a bank, and if you wanted to get organoids from our core, you can order them. You have to help pay, you have to buy them because we do have expenses in making them, but that is something that there are people around the country that are getting these from the, we might have a culture that you don't have that could be interesting. And I'll stop there and take questions. I think I may have gone a little longer than I expected, but there's still some time for questions. And thank you again. If you can help me with chemokines, let me know. Okay, thank you.
How this works from here. That is speaking of yeah. Hello, hi, yeah, I didn't talk here. Hello. I've got a question regarding this polymorphism you showed with the T1 and T3 particles. Is it understood what drives the equilibrium between these two particle types? And could this potentially be used as an antivirus strategy to drive the formation of the wrong particles?
Yeah, so when we made virus-like particles, occasionally we would see T equal to 1. Most of them are T equal to 3. We were never able, I mean, maybe we probably didn't spend enough time. I'm sure if we really wanted to focus on that. So we were never able to get enough T equal to 1 particles with VLPs made using the baculovirus system to really do a lot with them. We showed that they would bind. They bound to antibodies that we had. They don't have the genome inside them. But we really didn't do much else with them. And we have very, very few in these cultures. And so we don't know a lot about them, but that's a good idea.
So it could then be that the genome defines the geometry, that interactions between genome and protein lead to the formation of the T3s, which would bring me to my next question then. When you showed us this nice mechanism with this dynamic movement that leads to the egress, is there a possibility that potentially interactions with the genome on the inside or differential interactions at different points in the capsid shell could make a contribution to this mechanism at all?
I think that's very possible. So the genome is interesting. I didn't say this. It has a VPG that's attached at the 5' end, like maybe other single-stranded RNA viruses do. But what's interesting here is that VPG takes the genome, it binds to initiation factors. And so, which the other VPGs, to my knowledge, don't do. So we don't know what it's doing. But I have the Janum Dave, his thesis is looking at the interactions between VP2, which is a small structural protein I didn't talk about, but it's inside, we think inside the particles too. We're looking to see, we think that probably interacts with the VPG genome and brings those into the capsid. And I would not be surprised at all if that's really, yes, what makes the T=3 particle. But a couple more years to go.
Thank you so much.
Okay, Jan has a question. Jan, how do you
Yes.
say? How do you say your name?
It's Jan. I'm
Jan, sorry. Okay.
from the Netherlands. No, it's a fantastic presentation. I was wondering for sort of the model of like inducing membrane wounding and then having sort of the virus induce the wounding. For bacteria, sometimes it's sort of the other way around, that sort of the bacteria, they start attaching to sites, right, where the wounding is already occurring. And by this attachment, they enter. Maybe my question is, do you think there could be a receptor that get exposed by wounding, and which is what the virus then hones in on or uses to sort of specifically infect there? Or do you think about other mechanisms of how this membrane wounding is induced by the virus?
I mean, I think that's possible. We're really not going to know. There can be lots of ideas here. We think the receptor is in the rafts, but when the virus first binds to the histoblood group antigen, whatever that's attached to, it may be causing conformational changes in things that are in the raft. I don't know how it's going to work. I can't wait to find out, and I hope I find out in my lifetime.
And maybe another question, sort of the role of the pH in sort of the entry process. Do you think so? The part of it is right ceramide interactions and bile acid interactions, but sort of in isolation, the pH can induce it, can modulate conformational changes in the virus, or it could interact its interaction with a proteinaceous receptor. Do you have any sort of clue? Of course, right, we do a lot of our tissue culture with a pH of 7.4, which is not very physiological going through the small intestine. Yeah.
So, I mean, we looked at the effects of pH on at changing the media, and we didn't see any in the experiments that we've done so far. But the fact that the pH, the raising and lowering of the capsid now, which we didn't know about when we did those experiments, we're going to go back and repeat some of those. We know that the pH coming in the endosome is important, but that's about all we know at this point. It's kind of, although we have VLPs, it's not purified infectious virus, and so working with stool is still a little tricky for some of these experiments that can potentially or more easily done with viruses that you actually can passage and purify from normal tissue culture cells.
Maybe one final question. You see this interesting difference between the small intestine and the colon-derived organoids. Can those help to sort of direct the search for a receptor to look at differential expression between those rather similar, maybe, in tissue culture model systems?
Yes. So our top candidate for the G24 receptor is not expressed in the colon. by Western blot. That's about
Mhm.
all I can tell you at this point. And so we're gonna, we do not have it cloned yet, but we're gonna clone it and try and put it in the colon and see if that will now allow us to grow the virus there. We're gonna put it in other cells too, but that's what we'll be doing the next year or so.
Very exciting. Thank you so much.
Mary, it's Raul here. It's good to hear a very nice presentation. So I imagine this has been a frustration for you for many years, why you cannot pass the virus from culture to culture. Do you have a hypothesis why, what do you think is missing on the culture? Because obviously in humans, the virus passes.
Yeah. So, I mean, I think restriction factors are a big part of it. So when we knocked out STAT1, at least we got the G2/3 to spread, and we could go maybe 1 or 2 passages, but it doesn't go past that. So I mean, one hope is that by knocking out these other restrictor factors that we know are present in the STAT1 knockout, or we actually have a line that we made, the J4.2 line, which was a secretor negative that we put, that line grows many more viruses than we originally could grow. And again, it has a lower basal innate immune response. So we've just done an RNA-seq of that line, and we're now comparing, what's now still upregulated with infection in that most susceptible line in our J4, in our STAT1 knockout line, and in the pediatric lines we have, which also have a lower innate response. And we're hoping that that's going to really help us try to figure out what's the next restriction factor we ought to go after.
But do you think in people in a normal physiological condition, restriction factors in this tissue are reduced, or there is some pro-virus factor that is missing in the culture?
Well, I think in the acute infection, the restriction factors are very active. The G2/4 virus may be able to grow a little better. So the other thing is, I mentioned these cells are sloughing, they're being turning over every couple of days, and it takes a while for them, a day or so to be replaced. And it may be that the virus is also moving down the intestine while the small intestine's being replaced with any cells that were infected and maybe died and so forth. And so that may be part of, because this infection really, in a normal, healthy adult, we would infect people Thursday night. They'd be sick by Saturday, maybe usually Friday or Saturday. Sunday, they were usually better. And by Monday, most of them went back to work. So it's an acute, really quite an acute infection for many people. But if you're chronically infected, I mean, those are people that are all immunosuppressed. So obviously the immune system is what also helps clear the virus. And we don't know where the virus is located in people that are chronically immunosuppressed. We do know we got a biopsy from a person that had chronic variable immunodeficiency disease, so they don't make B cells and they don't have immunoglobulin. That person underwent a endoscopy and got a culture to us. We saw the virus in the enterocytes in the original tissue. When we made the organoid from them, or enteroid, the virus was gone. And interestingly, the virus from that person doesn't infect those cultures, but it infects some of our other cultures. I don't know why that is. That's a puzzle. Maybe that's a side story.
Yeah.
I don't know of other mechanisms of what prevents virus from passaging. I guess the main one, I mean, you have to have the receptor, number one, for sure. And you need to have, but these cells have the receptor, and you need to not have them be restricted. Maybe there are other things that I'm just not aware of.
Yeah. Is it possible the bacteria plays a role in the whole dynamics of infection here?
Well, in our cultures, there are no bacteria.
Right. But I mean, in people.
Oh, you mean if we had bacteria, it would enhance?
Yeah.
Yeah, we haven't looked at that. That's, I guess, there are examples of that with other, Julie Pfeiffer's work and other people.
Yeah.
There is somebody else at Baylor who is trying to look at bacterial supernatants to see if they might enhance. And so far, they really haven't found anything that would be a metabolite. I mean, there is the murine norovirus is a virus that grows in immune cells. And it also actually grows, depending on the strain, it grows in tough cells in the intestine. But I think that's a very different pathogenesis than the human virus.
Right. Thanks.
Mary, do you see a lot of evolution in the immunosuppressed people of the virus?
Yes. I mean, there are many papers. The norovirus field is not that huge, but there are people that have been working on that, and it's definitely evolving.
Thanks.
Are you asking for a specific reason?
Well, but it's just sort of that there's a lot of attention now, and on SARS-CoV-2 evolution in these immunosuppressed people. So especially
Right.
also with HIV, I just wonder whether that is a large concern.
Yeah, well, it is a concern. I mean, the main reason people, one of the questions is, where do these new variants come from? And so the main question that people were originally asking when they were looking at virus over time from immunocompromised people is, were they producing, unique variants? That doesn't seem to be the case. I mean, variants are happening, but it looks like it probably is kids where the new variants are coming from. And that, of course, their immune response, if they are having a primary response and don't have much of a antibody response initially, the virus is probably freer to make variants. So, I mean, that question is still an open question.
Thanks.
Okay.
Well, if there are no more questions, please join me on thanking Mary Estes for this wonderful seminar. Thank you so much.
Okay, thank you for
Thank you.
Okay, bye.
So